recording. Without further ado, I am super delighted to welcome Bruno Correia, who will be giving us the first of 3 guest lectures today on protein folding. So we are really privileged. We have Bruno, then we have Shin Boshu, and then Mohammed Al-Quraishi. So Bruno, take it away.
Yes, let's see. So here we go. Okay, so guys, it's really a great pleasure to be here with you today. Part of that I really appreciate about my job is to get to talk with students when people are still sort of like very eager to be excited about new subjects. And I hope that after this this very short 25 minutes that I have today to tell you about a few of the things that we're doing here in the lab, that you feel as excited as I do about studying protein structures. So here's the deal. Today we're going to talk about like what we commonly refer in the lab as surface fingerprints. And we've used geometric deep learning in order to study what the types of functional features that surface fingerprints in protein structures can actually teach us. The core of the subject that I'm going to tell you about here today as an engine, and that engine is what typically the experts in the field refer as geometric deep learning. And ultimately what geometric deep learning is basically this concept of studying high dimensional types of data where you have very rich data structures and that you can learn from them. So let's say that, as you know, deep learning has been doing like, a lot of like progress in many different fields, but perhaps the one that is the most noticeable is in image classification, right? So if you think about image classification is actually a pretty like lame type of data set. And in biology, we actually don't have a lot of like, data sets that look like images. They're generally much richer. They have sort of like time dimensions. They have other types of dimensions that we can put in each plot. And so geometric deep learning tries to work on that realm where we have much richer and high dimension data sets. And I will give you examples of exactly what these data sets look like. So I'll start just by giving you a brief introduction into protein structure and function, and then we'll go to how we can study this surface fingerprints on protein structures. And then I'll tell you a little bit about design if I have the time. If I don't have the time, it's also okay. Okay, proteins, right? Here's the deal. Proteins are encoded in our genome, and generally they're basically the workhorses of life. So we do get these codes encoded in our genome in terms of their DNA base pairs, and then ultimately the cellular machinery will translate this code into amino acids. One of the most amazing things about these molecules is that typically their function is related to their three-dimensional structure. So these polymers that you see here represented in a very cartoonish way will adopt a three-dimensional structure, and then these can actually perform molecular functions in cells, right? So they can do catalysis, so chemistry. They can do mechanical functions. They can do binding and recognition, and they really control all the types of cellular behaviors and organismal behaviors that we have. Okay, some ideas about protein function. So I'm sure that many of you right now are eager to have anti-SARS-CoV antibodies, right? Well, guess what? Here's the structure of an antibody. They all look like this, and this is one of the most how should I say, popular molecules these days in terms of what we would like to have in our body to protect us against these pandemics. But you also have other proteins like, for instance, storage iron, like heme, transport. So proteins that actually pump ions against gradients. Same thing. Same idea here. Communication in between cells. Rigidity, mechanical properties in cellular entities are also done by protein structures. And ultimately, one of the most amazing things is chemistry, like in terms of catalysis. Okay, so I will preemptively address a common point that I get asked, which is the work of studying protein surfaces been addressed by AlphaFold? And the reality of the fact is, and the answer is no. So what AlphaFold has done, and I'm sure all of you are aware of AlphaFold. You might not know exactly what a structure of a protein is, but you know what AlphaFold is. And what AlphaFold has done is really to solve, at this point, as far as we can tell, this long-standing challenge of predicting structure from sequence, which until now has been one of the outstanding challenges in molecular biology. And really, you can see here, so now what I'm showing you is the superposition of two modeled sequences or models that were created by AlphaFold in comparison with real structures. And you see that this is, these predictions are absolutely incredible. However, function prediction problem is a different thing. And we like to use structures in order to predict which kind of functional fingerprints they have in their structural, in their surfaces. And for instance, I will give you two examples here that a lot of, let me just connect this. Yeah. So for instance, one of the things that is the most important in order to ensure that cells work properly is really this idea of how proteins interact with one another. And here I'm showing you a network of, or several networks, in fact, of the protein-protein interactions that have been characterized in different organisms. And this is still something that is, in my particular regard, a lot harder to predict than the structure, although predicting a structure was still pretty hard and is still pretty hard. Or also, for instance, thinking about how proteins actually interact with other metabolites in cells. So all these things are very important for having a full understanding of what these molecules do in cells. Now let's go more towards why is it important to study structure in order to understand their function. And so here I take this quote from Francis Crick, right? And basically, already by the time that they were studying the structure of DNA, they were already sort of like with this cliche of, okay, well, if you want to understand function, you certainly should study structure. And when we talk about studying protein structure, there's many ways that you can study protein structure. And you can either represent protein structure, so the strings of amino acids that I told you about initially, as in a form of what their secondary structures is. So sort of like, this is more of an architectural definition of what the molecule, what the three-dimensional shape of the molecule is in once it's folded. Or you can have more sort of like a graph where you have the different building blocks represents as sticks. Or then you start getting into different types of representations. For instance, you could make the parallel that, the atomic representation of a protein is no less than a point cloud. And then, even a higher level up is this idea of representing proteins as continuous molecular surfaces, as I'm showing you here. And this will be the type of data that we are going to be working in the next slides that I'll be telling you about. Now, what is interesting about thinking about protein surfaces? One of the things that is interesting is that a lot of these surfaces can actually have dissimilar sequences, so different sequences. Also, they can come from different architectures of the protein, but yet they might have, or they can have, a similar function, which basically takes us to this idea that many of these similarities that we observe in proteins are actually only detectable at the surface level rather than the architectural level or at the sequence level, which is typically what you do. So for those of you that are interested, that are, used to study protein sequences, the first thing that you do when you have a protein sequence is, you blast it. And you blast it and you ask, okay, are there any other proteins that look like this protein? And once you understand if they look like this protein or not, then you say, okay, well, if my protein looks like this one, maybe it's doing similar things. But here the concept is a bit different, is if my surface looks like this, independent of this sequence, is it doing a similar thing? Okay, very good. So I like to do this, this analogy. Studying protein surfaces is just like studying people's faces. When you learn enough about patterns that you see in people's faces, you can tell many things about their, where they come from, their state of mind. Same thing for proteins. Or at least that was the hypothesis that we had. So for this, we of course departed from what the traditional deep learning is, which basically works more in a 2D type type of data sets, more in terms of images or acoustic signals. And we went more towards this idea of geometric deep learning, which is clearly much more suitable to the problem that we have, right? So which is more of a three-dimensional idea. And so let me tell you what the engine that we're using actually works like. And so the prototypical objects for geometric deep learning are graphs, and I'm sure you're all very familiar with graphs, or surfaces. Of course, that these surfaces can also adopt different types of data structures, and these different types of data structures can be volumetric, or it can be, for instance, point clouds, or it can even be sort of like meshes. And meshes are just basically triangulations of your surface that you can then work with. And so the mesh representation was exactly what we've used for to study proteins. So we're going to focus on this one. Now that you have this mesh, so you see that you have the nodes and you have the edges connecting each of the nodes. And now we can create our fundamental data structure that we're going to use to study also proteins. Here, first I showed you a rabbit, but that's essentially what we're going to use is this idea of patch. And basically what these patches are these data structures, which are subsets of a mesh. And then in each node, we're going to have several features. I will tell you what they are. Right now, I'm going to keep it very abstract, but you can have like vectors of features in each one of these nodes. And you will apply local geodesic polar coordinates in this case, where you will define which kind of radius you want to have and you want to reach around this mesh. And then, one of the biggest innovations here, which the innovation was that we applied it to proteins, but it already had been applied to other things by our dear colleague, Michael Bronstein. And basically what this data structure refers to is how you can actually set local weights for each one of these Gaussians, which Michael likes to call soft pixels. And basically what this is that you will give a learnable weight to each one of these areas of your patch, and then you will work from there. And then after this, you actually apply a spatial convolutional filter to this data structures, and again with two learnable parameters that you see here, both the G and the U are learnable, and essentially after this convolution, you boil down this information of this patch into a vector. And of course, that then this vector will have different types of lengths. This is something that then you have to work in order to improve your performances. Okay, so let's see. How did we use this for proteins, right? So we started with three-dimensional structures of proteins, transformed those structures into meshes, and then segmented it into different patches, which you see here. So just the geodesic patches that I've shown you. You see the nodes of the different mesh structures that we have here, and then you also have here an idea of that radius. And then we started putting attributes into it, and I think this is one of the most exciting parts of our work, because it was when we started actually sort of like thinking, okay, so in terms of if we want to learn about the chemistry and the structure of these protein structures, what should we encode, right? And ultimately we settled down to two different classes of features. So we encoded into each one of these points within these meshes different geometric features. So shape index, which tells you about the topology of the surface itself as the same thing as distance dependent curvature. And then on top of it, our biochemistry heart started speaking a little louder, and we started encoding chemical features into it. So hydrophobicity, so how hydrophobic is a particular node on this mesh, what type of electrostatic features it has. Then together with this, we encoded a polar coordinate system. So now each point within this mesh knows its orientation relative to all the others. And then we have we pipe all this data into the geometric deep learning approach, and we train different fingerprint descriptors for application-specific layers. And now the next part of the talk is to tell you about what type of applications do we have.
So okay, Bruno, this is phenomenal. I love this whole concept. If you don't mind going back to the previous slide. Now, how do you connect all of that to the sequence? Basically, right now, how we encode the surface.
Key question. This data structure has no perception of sequence. We are just really studying patterns of atoms and of chemical properties in the patterns.
So that's unrelated to the protein folding problem.
Absolutely.
But that's related to docking. It's related to sort of if you have things that are near each other, how do you go from that? But basically, if I have a molecule, you won't be able to tell me its structure, but you'll be able to encode its structure and learn about its structure and find similar structures if you have the structure.
Absolutely.
Perfect.
Yeah. But I think that's a good point, and I absolutely have not made it like very clear. Indeed, this data structures that we're learning here, they're completely agnostic of and I have to tell you, that was actually something that was very interesting and very sort of like intellectual stimulating, because that was sort of like the whole idea is like, let's learn the patterns, which are in a way there could be sequence degeneracy in these patterns, because with different sequences we might be able to present the same pattern.
Yeah. And for example, with a tRNA that's mimicking, or a protein that's mimicking a tRNA for a stop codon, you'll be able to sort of map their structure regardless of what they're made of.
In principle, yes. That's the idea. So sort of like, in a way, we would like to learn just which kind of patterns are, and this is something that we're studying right now, exist across different structures, because perhaps we can learn from different functions that this proteins might have, just because they bear these patterns in their surface.
I love it. Carry on. I don't want to interrupt.
Okay. Yes. I don't have the time. I will not have the time to go over all the materials, but that's okay. I'll stop whenever we need to stop. Very good. So applications, right? Why are we interested in learning this type of properties of structures? And the reason is because they will help us to learn more about particular structures that we might not know about, right? So some of the questions that we're interested in is, could we actually, for instance, classify better pockets that are present in proteins in terms of understanding which kind of cofactors or metabolites do they bind? Again, no perception of what sequence is in these pockets, but just do they have the features to bind particular ligands? One thing that is a little bit more, I guess, in a way abstract, but very, very interesting to us, right? In protein structures, are there sites which are more likely to engage other proteins, even if you don't know what other proteins there are? Okay, so sort of like there are just surface patches that are more for protein solubility, other surface patches which are more for protein interactions. And then a more common type of exercise is this idea: can I predict which type of structure or which type of configuration you can obtain from two docking proteins? So can I predict the docking configuration of two proteins using surface fingerprints? Very quickly, we compared our predictor in terms of the specificity of pockets to other predictors, and ultimately what we saw was that Massive did perform on par with other types of pocket specificity predictors. One question that we really like to do in this particular studies, because by heart we're biochemists, and we really like to understand, okay, but in order to predict this type of specificity, what questions can we ask to these networks? Is it the chemistry that matters more? Is it the geometry that matters more? Is it both, right? And so we started doing ablation studies to our network and trying to understand, okay, if we only encode chemistry, how well do we do? And in this case, we only do 0.65 in terms of rock AUC. But if we only have geometry, clearly we're not doing as well. So of course, this means that geometry by itself doesn't encode a whole lot of information, but chemistry encodes a bit more. But then when I get both together, then things look a little bit better. Good. So then, in terms of accuracy to try to predict which patches are more likely to interact with other proteins, here, I think we did a good advanced. It wasn't as sort of like, close range of what we had before. But we developed this network that is called massive site, and we compared it to other proteins. And here we can certainly predict, um, given a particular protein surface, which sites are more likely to interact with other proteins. And if you compare here, these differences in terms of rock AUCs are certainly much more noticeable than what we had before. So that was interesting. So then, in terms of the docking problem, what we did here was basically, I will take you very quickly through this. We basically do a fingerprint scan in terms of all the patches that are likely to interact with other patches. We select the most promising fingerprints, and then we have a stage of alignment, and we re-rank. And I just want to bring up a point here, maybe in this slide, where we compared massive search, which is basically a docking program, into one of the best docking programs that exist in that are available in the literature. And so this is ZDock and ZRank. And I think, something that is very striking about these results is, depending on how you study the results and how you do the selection for your number of successes. So let's say that if you have a perfect predictor, you would have 100 here and 100 here and 100 here. But as you put different bars of selection, your numbers go down. But if you see these numbers, there ultimately we can get the same number of right solutions as the best predictor. Now, what is interesting about it is that we can do it, 4 orders of magnitude faster. And the reason why we can do these computations 4 orders of magnitude faster is because we're not doing computations in the 3D space. We're really only doing computations on 1D space, on vector distances. And by doing just computations on vector distances, we can speed up and basically, let's say that we can now dock all the proteins in the proteome all against all, and do it in a very short amount of time, while ZDock would not be able to do this. So this was something relatively, exciting for us, because now it speeds up the process a lot. And so perhaps I only have like 3 minutes left, so I will not tell you about how we use this for design, but I want to tell you about how we actually went in the next generation of massive, and that we called D-massive, for the differentiable massive. So if you followed up all the story, basically what we were doing before was to basically create a mesh of a protein, and then we were using essentially handcraft descriptors to map electrostatics, to map geometry, to map all those things. So all those things were basically done on the top of the mesh that we have already computed for the structure. So what we thought here was, okay, well, what if we would get rid of those pre-processing steps, and we could actually have an end-to-end creation of a point cloud that describes the surface, but also a computation of the geometric and chemical features. So the geometric wasn't that hard, but the chemical features was actually relatively hard, because basically what you're saying here is that you can compute the electrostatics of a molecule just by having the surface points. And so that's what we did here by developing this D-massive. And so some of the differences that you see here is that there was a lot of pre-processing steps that you see for the old version. And now what we have is essentially a full differentiable network that it was trained also in order to compute the electrostatic properties of this point clouds. And so when you see that, for instance, if we compare what we could consider the ground truth that we were using before and the D-massive prediction, for instance, in terms of the electrostatic potentials of this protein surface, they're actually not that different. We see some differences here in this pocket, but honestly, they're kind of like on par for what we need for these predictions. Okay, so with 1 minute left, what should I tell you?
Tell us a little bit about the design part. I think this is very
okay. A little bit about the design part. So let's see. Okay, the design part is always an exciting one. It will probably take me more than 5 minutes, but let's see. So we took this protein, and this protein is called PDL1. What this protein does is essentially Very important cancer for control, very important target for controlling the activity of T cells, okay? And what we wanted to do, and so there's a number of monoclonals that target this protein. But all we wanted to do is, okay, can we actually use massive to target this protein and design it, and the novel protein-protein interaction? So we used massive to predict a site that would be more prone to be targeted by design molecules. And then we extract the target surface fingerprint. Due to the speed that we have to do these docking calculations, we could actually create a large fragment, you know, with 100 million fragments of protein motifs that we could dock into this site. And then we docked motifs into this site, okay? And so when ultimately what you get out of these calculations is what are the most likely motifs to interact with this site that we're interested in to engage in. And then, you know, for reasons which are related to the experiments that we need to do in the lab, which we typically use a lot of yeast display to screen many different types or large libraries. We then transfer this binding site into globular folded proteins. And now I'm going to jump like very quickly just to show you the key results, okay? And the key result is this, okay? Typically, if you are a computational protein designer, what you have to do in order to convince people that your result, that your prediction is correct, is you have to solve an experimental structure. What I'm showing you here is what we predicted using massive and an overlap of the experimental structure that it was solved in the lab. And you see the experimental structure in pink, and you have the model and the seed model that we created using massive in orange and yellow. So now the root mean square deviation of this complex is around like 1 angstrom, which basically means that all the atoms are on top of each other, which means that the prediction matches exactly what it should have matched. And besides the structure, this is also a binder that binds to the PDL1 protein with 19 nanomolar affinity, for those of you that are more into the affinity constants, which basically means there is a high affinity interaction. More importantly, and this is absolutely the last slide, we did not recreate a motif that it was known in nature, because we also knew that there was the native ligand, which is PD1, that binds to PDL1, looks like the green structure that you see there. But once we actually overlap the structure that we designed, it looks nothing like what the native structure shows. Meaning that, you know, we enlarged the space of binders using surface fingerprints. With this, I think I really need to be respectful of the time.
Thank you so much. So we also already have Jinbo here. So everybody give Bruno a big hand. And then Jinbo, please start sharing your screen. So thank you. That was phenomenal. I truly wish we had a whole hour and a half. So.
No problem. Thanks a lot.
Thank you, Bruno. Bye-bye. You're welcome to stay, of course. And the question I always ask everyone is, if students are interested in doing projects in this area, would you be interested in potentially advising them?
Would love to. Yeah, it would be great. Yeah. And I can tell you that if you want to then cross the ocean, Switzerland is really beautiful.
Sounds great. Thank you so much. Bye-bye.
Ciao.
All right, everyone, please join me welcoming Jinbo, who will be telling us about protein structure prediction by deep learning. So Jinbo, take it away. You're muted and your video is off, but we can see your pointer and your slides.
Okay.
Perfect.
Okay, great. Yeah. Okay, yeah. Thank you, yeah, for giving me such an opportunity to talk about
your head. Can you tilt the camera just there? Perfect.
Okay. Yeah, not sure why my slides. Okay. Yeah. Okay, so yeah, I'm going to talk about how to use computer to predict the tertiary structure of protein from amino acid sequence without using any experimental data. So this is a very old problem, has been studied for many years, and the progress was not very good before. So many methods have been developed for protein structure prediction. Broadly speaking, we can divide all these methods into two big categories. One is template-based modeling, and the other is template-free modeling. So by template-based modeling, we mean we can use some source structures in PDB as templates to build a model for a protein sequence under prediction. So before, this method is actually a very reliable method as long as you can find some good templates. And it was the first choice for protein structure prediction before. Only until recently, now the template-free modeling now actually for many targets, for many proteins, the template-free modeling now has better accuracy than template-based modeling. So template-free modeling was used before, mostly when you couldn't find a template in PDB. But now, even if you can find some templates in PDB, you should also consider template-free modeling as an option, unless the templates are very similar to the sequence. So before 2016, when we talk about protein structure prediction, usually means we will need a lot of computing power. And even so, the success rate for template-free modeling is very low, even for only small proteins. And for very large proteins, then it's almost impossible to get a good prediction. By the way, so in this lecture, I will mostly focus on the template-free modeling method. So here is a very popular old method for template-free modeling. So this method is called fragment assembling. Here is so this picture show you the major idea, the rough idea of this method. So basically, given a protein sequence under prediction, we cut the sequence into some short segments. So each short segment has, say, maybe 9 or 11 residues. Then for each short sequence segment, we go to PDB. We search the sequence segment among the PDB to find some fragments for each short segment. Say, for example, for each short segment, we can find about 200 possible fragments. So the structure of this short segment may be similar to some of the fragments. Then we can run sampling. We sample each fragment from one for one short segment and assemble all these fragments together to form a complete conformation. And then, of course, we calculate the energy function and keep the conformation with favorable energy. So this is the basic idea of the fragment assembling. But in order for this kind of method to generate some good conformations, then we have to do lots of sampling. Yeah, we need to sample maybe 100,000 or even 1 million conformations for even small proteins, and it needs a lot of computing power. And so if it will take a very long time if you wanted to, like say, sample a protein with more than 150 amino acids, and the success rate is very low. So this is the previous popular method for template-free modeling. So only until recently, so we have so the community comes up with a new strategy for template-free modeling. So this method is totally different from the fragment assembling. So given a protein sequence, we first we run homology search. So we search a sequence against the large sequence database and find some sequence homologs and build a multiple sequence alignment. So by the way, so the popular fragment assembly method like Rosetta also will do the homology search, but this new method uses the sequence homologs different from Rosetta. So we build a multiple sequence alignment. Then from this multiple sequence alignment, we can actually some useful information. Like for example, we can get the sequence profile. Sequence profile just so encodes the information, the conservation information at each position of this protein. And then we also do some. We can also do some co-evolution analysis. So the co-evolution analysis will identify those positions which are likely co-evolved. Then we feed this information into some machine learning models, especially now deep learning models. From the sequence profile, we can predict the local structure, like the second structure or the facial torsion angles. And then from the correlation information and also the sequence profile, we can predict the interaction stress between any two residues, especially, let's say, we can predict the contact or distance between any two atoms or residues in the protein. Then we feed this predictive information, the predicted interaction matrix, and also the predicted local structure into a optimization engine to build the 3D coordinates. So this is a relatively new pipeline used for template-free modeling. So from here, yeah.
So for the multiple sequence alignment, is this based on the whole protein, or are people also trying to align smaller parts of the protein at the level of domains?
Yeah, so this is a very good question. So different people use different strategies. So for example, so for some programs, they require at least 50% of coverage. But so in my practice, I don't have such a requirement. So for me, sometimes I also cover, maybe for a small domain, I also want to have some coverage. So usually I require, as long as the number for covered residues, say, is at least 60 or say 80% of the sequence, the query sequence, then is fine.
Now, the other question is about evolutionary distance covered. You want many, many sequences at distances that are very, very far and yet preserve the structure. How often do you get that, and how hard is it to find a sweet spot of evolutionary space?
Yeah, this is a very good question. So I think this is still a unsolved problem. So yeah, to determine which sequence, how much should we use? So right now, we just use this very simple method. We just use this e-value cutoff. Say we set e-value to be 10 to minus 3. But I don't think this is the optimal solution. So I think a better strategy is you can use different e-value or different criteria to generate different multiple sequence alignment. And for each one, you can generate a model. Then you take the model with the minimum energy. So this will give you, generally speaking, will give you better 3D modeling.
Got it. Thank you.
Yeah. So of course, yeah, if you can find a human experts, if you can find experts who can fine tune the multiple sequence alignment for you, I think it will also improve the final 3D modeling. Yeah. So this is a relatively new strategy for template-free modeling, and so the key component here is how we can predict the interaction matrix. So the local structure is not so important. If you have a good prediction, it will help slightly. But the most important component here is how we can predict the interaction matrix. So we can explicitly model the interaction matrix using contact matrix or distance matrix. So here is the definition of the distance matrix and contact matrix. Suppose we have 4 residues here. Then we can calculate the Euclidean distance. Then we write down all this distance information in a matrix. Then this matrix is called the distance matrix. And of course, the distance matrix is very complicated since the number of entries is square with respect to the sequence length. Then we can simplify. We can just discretize into a contact matrix. So we use 8 angstrom as a cutoff. So if the distance is less than 8 angstrom, then we write down 1. Otherwise, we have 0 here. So in this contact matrix, the 1 indicate 2 atoms or 2 residues that are close to each other in the 3D space. So in the contact matrix, the number 1 is linear with respect to the sequence length. So it's simpler than distance matrix. So before, the research in this community mostly focus on the contact matrix because the precision of contact precision was very low. So the community only works on this simpler problem. But only until like the 2017-18, then since now we can do contact prediction much better, then we move to the distance prediction. So the contact matrix has been studied for many years, and the precision was about 20% for a long time. And so even like say many people believe the contact prediction is as hard as the protein folding itself. But actually, about 10 years ago, so now about 10 years ago, the contact prediction can be gradually becomes easier. So we can do contact prediction much better than before. So here are some ideas that are useful, very effective for the contact prediction. So the first idea is a global statistical method for coevolution analysis. And then the second one is deep convolutional residual neural network for prediction of contact or distance. And finally, is a transformer-like network for the contact prediction. So I'm going to tell you explain to you these three ideas. So this picture just explain what's coevolution. So look at here. Suppose we have so look at this two residues. Suppose one mutate, then it's likely the other one will also mutate in order to maintain stability of this structure. Then when we look at the sequence, the multiple alignment, then so this this position corresponds to this column here, and this one corresponds to this column here. So when we look at these two columns, then usually these two columns, they are highly correlated. So this means when we find some columns in the multiple alignment, if they are highly correlated, then likely the corresponding residue will be in contact in the protein structure. So based on this observation, then we can go to analyze this multiple sequence alignment. So we can develop some methods to identify those co-evolved residues to do the contact prediction. However, this problem is very challenging. So very long time ago, the researchers in this community use mutual information to do this, but mutual information just introduced too many false positives. Only until about 10 years ago, we have a better method, like the so-called the global statistical method. So global statistical method works much better than the mutual information. But the issue with this global method is it will need a large number of sequence homologs in order to get some good prediction. Otherwise, this global statistical method will fail. And then, so in this slide, so I show you two pictures. So this picture shows you the distribution of the protein families. So the top one shows you the size distribution of those protein families with at least one solved structure. So you can see the median size is about 680. So this picture was was drawn in 2017, but maybe now the median size is larger. This is the top picture. So you can see here, for those protein families with solved structures, they usually have many sequence homologs. And the bottom picture shows you the size distribution of those families without any solved structures. So you can see here, the median size is only about 100. So it's much smaller than those families with solved structures. This means the global statistical method usually only applies to those families with solved structures, but does not apply to those families without solved structures. Yeah, so this means the global statistical methods are not very useful in practice. So we need another method to handle those families without many sequence homologs. So this is why we wanted to develop a better method to do the contact prediction. So in addition to the coevolution analysis or the global statistical method for coevolution analysis, and then another line of this community is using supervised learning to predict contacts. So the coevolution analysis can be treated as the unsupervised learning, and so because it doesn't use any structure information, so it's only based on the multiple sequence alignment. And the supervised learning, so we will make use of both sequence information and also structure information. So now we should use, so this method will use structure information in the PDB. So here is a typical procedure adopted by previous supervised learning method. So suppose we wanted to predict the contacts for a sequence here, then for any two residues I and j here, we take some sequence features for I and also some sequence features for j. And of course, we can also take some pairwise features for I and j. For example, we can use the mutual information or even the coevolution signal derived by the global statistical method. Then we can fit this kind of features into a machine learning model and then to predict if there's a contact between I and j or not. So this is the typical procedure used by previous supervised learning method. And for the machine learning model, we can, many different machine learning models have been tried, like the neural network, support vector machines, random forests, or even deep belief networks. But still, even the DBN was used in, say, 2012. So the precision is still not very good. Still is between 20% to 30%. And so the DBN method doesn't have any advantage over the traditional neural network method. So the major issue with this kind of procedure is, I would say this kind of method is a local supervised learning method, because when they do the prediction for two residues, they ignore a large amount of information in other residues. So they only use information surrounding these two positions, and it doesn't use information in the whole protein. So this is a major issue with this kind of method. Just think about, say, the protein. So a protein has many residues, and all the atoms, they interact with each other to form a stable shape in cell. And if you just take two residues out of this protein, then it's unlikely this two residues will form a stable shape. So in principle, it's impossible to get a good prediction if you only use information from two residues. So this is a major issue with the previous supervised learning method. So to deal with this, then we developed a new method. So this method will predict all contacts in a protein simultaneously. So instead of just looking at the information surrounding two residues, then we will look at information in the whole protein. So this method is called the fully deep convolutional residual neural network. And so in this work, so we first show, so by using deep learning, we can significantly improve the contact prediction. If you want to know details, you can check out our paper. So actually, for some very hard protein test proteins, we can triple the precision, the contact precision by using deep learning. And then we also find out, so by using the predict contacts, we can fold some very large and hard targets in Cameo. So Cameo is an online benchmark, so it's different from CASP. So CASP is a offline benchmark, and Cameo is online benchmark. And more interesting. So we find out, so this model works very well for membrane proteins. So although in PDB we know we don't have many solved structures for membrane proteins, but actually we don't have to train our deep learning model by membrane proteins, and it also works fine, works very well on membrane proteins. And also the model trained on single chain proteins also works very well for the complex contact prediction. Yeah, you don't have to train. Of course, if you retrain, say using complex, it will improve. But even if you don't retrain the model, it still works fine. Yeah, works well. So this is the advantage of this this method. So now I'm going to tell you some technical details about this method. So here are the key ideas. So as I mentioned, so we wanted to predict all contacts simultaneously. So we model the interaction between two residues or two atoms. So we model them as an image. So each atom pair or each residue pair is treated as a pixel in the image. Then for each pixel, we wanted to predict a neighbor. So we want to predict the interaction strength, right? So we predict all contacts of this protein simultaneously. Then we can formulate this problem similarly as an image semantic segmentation. Of course, there's some difference, but it's kind of similar to the semantic segmentation. Then by this way, then we can apply very deep convolutional neural network.
Jingo, in terms of segmentation, do you mean spatially coherent? Because the thing with images is that the patches make sense in some, sort of place where convolution can work, as opposed to having very distant aspects being related through the same column, for example, but not necessarily local in XY space.
Yeah, so here, so in this formulation, mostly we model those, let's say, structural motifs, like say, we model those residues which are close to each other in the space, in the slit space. Yeah, but of course, we can also model the interaction, say, in one column or one row. Yeah.
Yeah. Because sort of how a particular amino acid falls will affect the entire column.
Right, exactly. So yeah, so this is the improvement over this model. So actually, yeah, last year there are some improvement on this, yeah, by model this kind of interactions. Yeah.
Great, thank you.
Yeah. Yeah, so by using this kind of image to model a protein structure, then we can predict all contexts of a protein simultaneously by using the convolutional residual neural network. And so here, assume everyone knows what's convolution, and then here I just want to mention the residual. So residual is very important. Yeah. So by using residual, then it enables us to use very deep convolution network. If you don't use residual, you can use at most say maybe 10 convolution layers, and then but the 10 convolution layer is not enough. It is far away from enough. For example, now we use 100 layers, which is much better than 10 layers, and now other groups even use, let's say, DeepMind, they use 600 layers. Yeah, so it looks like the more layers, then the better precision we can achieve, and without worrying about the overfitting. Yeah, so this is the residual neural network. Then here, this slide show you the input and output, as I mentioned before. So we have a multiple sequence alignment here. Then we can get two kinds of features. One is sequential feature, like the sequence profile, and the predicted local structure. The other one is the pairwise feature, like the mutual information. Then we feed them into the convolution network to get the contact prediction. And here is the network architecture. So since we have two kinds of features, and we use two kinds of residual network. So this is for sequential feature. So we feed the sequential feature into the 1D residual network, and then we convert them to the pairwise feature. Then we feed the pairwise feature into the 2D residual network, and finally we got the prediction. Of course, you don't have to use the 1D. The 1D is not so important. So you can directly convert the sequential feature into a pairwise feature, and then just feed them into the 2D residual network. So this is the network architecture, and so yeah, the 2D residual network is the most important. So yeah, the more layers are usually the better. And so here, I show you some results. Like here, so this is the ranking in CASP 12 2016, and 2016 so I'm the only group who used the deep residual neural network. And so although we haven't fully implemented this method during the CASP 12 season, but still our method was ranked first back then. And
with
then
a very large difference too.
Yeah, so there are some differences. Also, difference is not very large, but there are some difference. So this is our average rank, and then just the second one, the average rank, the second one. Yeah.
Wow.
Yeah. So this is the CASP 12, and in CASP 13, so many groups re-implement this algorithm, and this is the ranking of the contribution in CASP 13. And so you can see now many groups did very well and much better than the traditional method. So this shows you the traditional neural network, the accuracy of the traditional neural network. So you can see the difference between the traditional method. And this one is a global statistical method for co-evolution analysis. So you can see for CASP hard targets, the global statistical method usually does not work because many CASP targets just simply just doesn't have so many sequences. Many of them have like 10 or 20. Only 10 or 20 sequence homologs. Yeah. And so this shows you some top method, the contribution precision and F1 value, some top method. Like so, this is CASP 13, and this is AlphaFold, and this is my program RapX and Zhang group. So this is the F1 value, and so you can see in terms of F1 value, so there's no big difference between AlphaFold and RapX in CASP 13, and so this is the precision. Yeah, and of course, some, like say, this trivial zeta is after CASP 13, Baker group developed trivial zeta, and so further improve the precision. And then my group also further improve the precision after CASP 13. So yeah, on CASP 13 hard targets now, the precision can go to 80%, which is much higher than previous. So here I show you the progress on contact prediction in the past 4 CASPs. So in this picture, so X is the predictors, and Y is the precision. So each color represents one CASP. So you can see in CASP 10, right, so the precision is about 20. The best one is about 20%, And in CASP 11 is about 27%, so this is best precision. And CASP 12 is 47%. And CASP 13 is 70%, and now we can do 80% on the same CASP 13 template.
What's driving all of this? Is it just depth, computational power, or is it truly new architectures each time?
Okay, yeah. So this one in CASP 10 is by the traditional supervised learning method, and CASP 11 so it's a combination of the traditional supervised learning method with the co-evolution analysis. And in CASP 12, so half are from the deep learning method and half from the enlargement of the sequence database. And in CASP 13, it's purely from the deep learning method, from the better engineering, better implementation of the residual neural network.
Wow. And where do you think we stand? Do you feel that there's like tons of room still, or do you feel that we're kind of saturating that?
Yeah, I think for content prediction, I think by using transformer like the AlphaFold 2, they should be able to do better, than my method,
Wow.
Yeah, so this is the progress on content prediction. So you can see the content prediction has been significantly improved by using deep learning, and so since now the precision is 80%, so it's very useful, more useful than the previous. Like when it's only 20%, then there's almost no use, not useful at all. But when it's 70 or 80%, then it's very useful. So after the content prediction, after we found out the content can be improved significantly, then we move to do the distance prediction. So we actually we don't have to change the deep learning model. Still, we use the residual network. So we do the distance prediction. So we first, so we find out by using the predict distance, we can significantly improve the template-based modeling. Yeah. So we published this paper in early in 2018, and then later we move to use the distance prediction to do the template-free modeling. So after we have the distance prediction, then we need to build the 3D model. So there are two kinds of methods. One is use the distance geometry, which was originally developed for like for the NMR experimentalist. So they have an NMR measure, then they wanted to build a 3D model. So you can we can use distance geometry, but distance geometry actually is not very good for the predict distance, because the predict distance still has some the error are still larger than usually than the NMR data. So the other method is to do the energy minimization. So we can convert the predict distance probability to some energy potential, then we minimize the potential by like the gradient descent or confirmation sampling, and then we can build a 3D model. And here, so the gradient descent works very well and it's very fast. But also, you can also use confirmation sampling. And now, even if you use confirmation sampling, you don't have to sample many decoys. I think usually you just need to sample 100 decoys, then should be fine. Because the energy function is much more accurate than before. So this is the method to convert the predicted distance into the 3D models. And now I'm going to show you some results here, more results on 3D modeling. So first, we needed to prove deep learning indeed can generate new structures, right? Right? So we don't want the deep learning only simply memorize the training proteins, right? So then, in order to demonstrate this, we calculated the structure similarity between the test protein and our training protein. So in this picture, the X show you the structure similarity between the test protein and our training protein, and Y is the quality of our predicted structures. So you can see for most test proteins, the predicted structures has a much better quality than the structure similarity between the test protein and the training protein. So this means deep learning indeed can generate new structures instead of just simply copy from the training proteins. And then we also study how many sequence homologs we need, right? So this picture shows the relationship between the quality of the predicted model and the log of the number of sequence homologs. So you can see when the log value is larger than 3, then for most of them we can have very good prediction. So the log value is 3, then it means the number of sequence homologs is about 20 to 30 sequence homologs. So as long as we have dozens of sequence homologs, then we can get a very good prediction. Yeah. So this is this means we don't need so many sequence homologs. So if you use co-evolution analysis, just the global statistical method, you will need 1,000 sequence homologs. And then we also studied the importance of the co-evolution information. Even if we don't use co-evolution information, actually for many of them, for many test protein, we can still get good prediction. Yeah, for certain of them, the predict structure still has a better quality than the structure similarity between the training proteins. So this means deep learning indeed can learn some important sequence structure information even without co-evolution information. Yeah. So this is the results this is some interesting findings we got. Yeah.
So can you use all of these existing predictions as a feedback into a training set to basically start predicting with coevolution much more, a much larger space, and then use that as an input function to then get better sequence-based predictors that are not relying on coevolution?
Yeah, I think this is a good idea. Actually, someone is trying this. Yeah, he's trying to see. Yeah, so you can use the coevolution method to get some predict structure, and then use this structure to do supervised learning. Yeah. So this is some results, and then what's next? So the next step. So yeah, so after this residual network, then there are some very good improvement. Like say we can use sequence structure information better. Like say the Facebook developed some very interesting self-supervised learning method, and of course, in addition to Facebook and other groups, there are also several other groups did this. And then we can also use template information to, as I mentioned, so now actually for many proteins there are some good templates. So we can combine the template information with the deep learning to improve the modeling further. And actually my group started this idea in 2018, and DeepMind did this in last year. Then we also learn the sequence weights, because the sequence there are some redundant sequences in the multiple sequence element. So not all the sequences are equally important. So we can learn sequence weights in the multiple sequence element. And of course, we can also use a better deep network architecture, like say we can replace the co-evolution analysis by residual net, or just replace the residual net by the transformer network. So in terms of the network architecture, I think the most important is the transformer, right? So we can use transformer to replace the residual network. So Facebook started to use transformer to do the self-supervised learning a few years ago, but Facebook doesn't do the supervised transformer. And DeepMind uses supervised transformer like network, and it did very well. And of course, another trick is to do the almost to the end-to-end framework. So this will improve the performance also. And so for template information for some proteins is also important. So if we can find reasonable or good templates, then you should try to combine the template information with the multiple sequence alignment, then to improve the 3D modeling accuracy. And here I show you some examples, especially for some very large proteins. And if you can find a reasonable template, then try to use it. Yeah, I think it's important to use it. Like here, this protein has almost 500 amino acids and fewer than 200 sequence homologs. So it's not a lot of sequence homologs. So if you don't use templates, then by using deep learning, then this is the model. So so the domains, they are not well arranged. So for each domain is predicted very well, but the orientation, the relative positions is not correct. Then here is the model with templates. So we only use templates, but not the multiple sequence alignment. Then if we combine them together, then we can get a better model. So combine the templates and the deep learning, then we can improve.
So Jinbo, how much physics goes into that? I mean, it's amazing that deep learning gets so far, but can physics help you get any further, or is physics basically not useful at this point?
Well, so this is I think this is a very challenging question, and I don't think we have a definite answer to this. So Michael Fick, so who is a biophysicist, so he has some physics-based refine method. So he can refine our model. So he can refine my model produced by deep learning by using physics-based method. But he couldn't refine the model produced by AlphaFold 2. So yeah, depends on the model. So for some models, then the physical method can still help to improve.
Great.
But actually, for those models, now I also have a deep learning method to refine. I can also refine use deep learning without using physicals. Yeah.
Got it.
Yeah. Okay, so yeah, this is for one example. Then here is another example. So this example has this protein has more than 700 amino acids. So now we can do very well without using templates. By using deep learning, we can get a very good model for such a large protein. Yeah, because this protein has more than 13,000 sequence homologs. Yeah, for such a large protein. And then this is the model built with templates, but without any without sequence homologs. Then we combine them together, then we can get a better model. So we combine the templates and then multiple sequence alignment, and then use deep learning. Yeah. So yeah, these two examples show you. So for some very large proteins, yeah, if you can find reasonable templates, then I think maybe it's better to use it to combine it with the deep learning method. Yeah, to improve the model quality. And actually, in CASP14 last year, several groups has adopted this strategy, like AlphaFold 2, and also David Baker's group. They also use the template information. And then, so this slides I show you the workflow used by AlphaFold 2. So AlphaFold 2, yeah, using an even more simplified workflow than my method. Like here. So yeah, given a sequence and the prediction, AlphaFold 2 will find a sequence homologs and build a multiple sequence alignment. And then also find some templates if some good templates exist. Then we'll feed these two kinds of information. We'll feed the multiple sequence alignment and the templates into the deep network. So here you can see, so the AlphaFold 2 doesn't use the global statistical method at all. Just directly feed the multiple sequence alignment into the deep learning. So this makes things simple, and also make it is possible to do the end-to-end learning. And then the transformer will infer the interactions between residues. And then instead of to generate the distance or contact information, then this kind of interaction matrix will fit into another deep network, to produce the 3D coordinates. Yeah. So this is the workflow used by AlphaFold 2. And so so you can see here, there's no confirmation sampling, there's no gradient descent, right? So there's no energy minimization. So this is almost end-to-end framework. So the prediction error in the 3D model can be directly fed back into the input, into the multiple sequence alignment, and also into the template.
Now, what's preventing everyone from doing this? Is it just like massive compute power, or what,
Yeah. So like if you want to use a transformer, you really need, many GPUs in order to do the transformer very well. And the GPU memory is a bottleneck.
Got it. And what about training data? Is that also a bottleneck?
So according to DeepMind, they only right now they only use the solved structures in PDB. Yeah, they indeed use much more training data than us, but it's still feasible. So like say, we use a smaller training set. They use a larger one, but I think it's still doable. The major bottleneck is in how we can implement this transformer when we don't have many GPUs, right? Yeah. The other is DeepMind also hired one human expert, to fine-tune the multiple sequence alignment, to manually search for the sequence homologs, which are very important. Yeah. So even you can find, say, only 5 or 10 sequence homologs will make a big difference. Then you don't have any sequence homologs.
That strikes me as a bottleneck. Where what if we build a machine learning model that can choose better homologs?
Right. I think this needs, worth study. So how we can find better homologs, how we can find do better sequence alignment.
Yeah.
I think this is where we can improve further.
Yeah. Perfect. Yeah. All right.
So almost done. This is the final slides. I show you the progress on 3D modeling. So this picture drawn by John Mote, who is the CASP organizer. So show you so each curve represents one CASP, and of course the higher the curve, then which means the better quality of the we can predict. And so in this picture, X is so we arrange the tasks from easy to difficult. So at the at the right, so the most difficult targets. And the score is ranging from 0 to 100. So 100 is a perfect score. So you can see the gap, so you also see the progress on the most challenging targets. So from CASP 1 to CASP 12, so CASP 12 is in 2016. In CASP 12, although I started the neural network, but I didn't get a chance to apply the predictive context to 3D modeling. So the 3D modeling was still built by old methods. So you can see from CASP 1 to CASP 12, so in terms of 3D modeling, the improvement is about 17 points by using the fragment sampling and the convolution method. Then by using the neural network from CASP 12 to CASP 13 and CASP 14, then by using the neural network, we can improve the score by 33. Yeah, and then by using an end-to-end the transformer model, so the AlphaFold 2 can improve over the neural network by about 19 points. Yeah, another significant jump. So you can see so by using deep learning, in total we can improve the quality by 52 on the most challenging test targets.
Beautiful.
Yeah, but of course, in real world, actually, we don't have so many very challenging, so difficult targets. In real world, I would say you just look at the middle region, so maybe it's more realistic performance jump. Yeah.
Lovely. Thank you so much, Jinbo, for a fantastic lecture. I just want everybody to join me in really, really thanking you big time.
Okay, yes, thank you. Yeah.
Awesome. So we are very fortunate today to have a third guest lecturer who has been patiently waiting. So Mohammed Al-Quraishi, if you mind joining us, thank you so much for coming as well. And please do share your screen. It's truly a privilege to have all three of you today. And I think our students are just very grateful. So thank you so much. All right, take it away, Mohammed.
Thank you very much for the invitation, and it's great to see Jinbo and Bruno both here. It's nice to that we have this sort of virtual capability nowadays. So I think Jinbo actually gave a pretty good sense of where AlphaFold 2 currently sort of stands. I'm happy to answer any questions that people have on AlphaFold 2 because I know it's sort of of immediate current interest to a lot of people. I won't sort of focus on it too much in this talk. I'm going to focus mostly on work that we've been doing since then. But I'm happy to take questions in the Q&A or whenever, even during the talk, if anything arises about the specifics of AlphaFold 2 and how it relates to what I'm describing.
Beautiful. Thank you.
Oh, you're quite welcome. So I'll start out, and maybe this is slightly redundant with some of the stuff that Jinbo talked about, but just to kind of orient you a bit with the way I view the space in terms of how algorithm space for putting such a prediction has sort of evolved over the last 2 decades, really. And there's obviously many ways in which one can sort of conceive of this algorithm space, but I'm going to essentially kind of think about two main axes. One is how much information goes into a given algorithm, so how much sequence information, and the other is obviously the accuracy of the method. How well does it work? So if you sort of look back, kind of the earliest methods, dating back to like the 90s, essentially with Rosetta, these were sort of really kind of inspired by the idea that you could use a physics-based model, an energy function, combined with sampling to get at the lowest energy state of a protein that would presumably correspond to its native state. So in this particular kind of context, the inputs are basically just single sequences of proteins. And like I said, Rosetta, Itazza, and a few other methods were at least in part sort of physics inspired, not entirely so, but at least in part. Now, these methods did work on single sequences, but I think it's fair to say, even till today, their accuracy remains somewhat limited. They work better if you sort of incorporate additional information like structural templates, like Jinbo was mentioning. But nonetheless, I mean, without using any kind of additional sequence information, it remains quite difficult, really, to do well on this problem. Now, things really began to change, I would say, about 10 years ago, around the early 2010s, when this idea of coevolution really sort of came to the fore with the kind of basic notion that you build a multiple sequence alignment, right? And then you look at how residues co-evolve with one another. The initial approaches were sort of all unsupervised, so they essentially just tried to extract information using kind of sort of various Potts model type of approximations. but there was no sort of directed learning. This is just purely kind of coevolution. There's just purely statistical inference, but that typically involves instead of post-processing, again, like Jimbo mentioned, to actually fall the protein into a well-defined three-dimensional structure. And these, I think, were certainly better than the sort of purely physics-based approaches or quasi-physics-based approaches, but they were, but they didn't make a huge difference. It was an interesting idea. It seemed to be promising, particularly for membrane-based proteins and single membrane proteins that are generally, we just don't have that much structural data on them. And because these were unsupervised, it actually worked well, irrespective of how much structural data we had. But I think it's fair to say that in the early 2010s, at least, they didn't really make a huge difference. That began to change actually with Jim Wuzu's work with RaptorX in around 2016, which was sort of, I would say, kind of the first approach to really bring deep learning. There were neural network-based approaches, fairly old ones included, but sort of real kind of deep architectures, leveraging kind of all the innovation that's happened in the last decade, really kind of started with RaptorX in 2016, particularly with sort of a residual network architecture. And I'm sure Jim would tell you all about it. That was followed by another architectural sort of innovation where in the kind of the RaptorX original formulation, we were still using this idea of binary contacts. So whether two residues are essentially in sort of a binary state of being adjacent, or not really adjacent, they're usually within 8 angstroms, but some notion of distance proximity. And those constraints, I should say, that were then used into using the physics-based engines to actually follow the protein. But RaptorX 18 actually sort of changed this idea and said, okay, what about if we predict distances, continuous distances, instead of these kind of binary contacts? It was a simple change, but it had a fairly substantial change, and was sort of perfected, I would say, with AlphaFold 1, which sort of took this idea and sort of engineered it really, really well into some much deeper networks and that sort of thing. This question came up earlier. Just a much better kind of neural network engineering. And that really sort of allowed the field to advance quite a bit and sort of generated a whole bunch of kind of, I would say, sort of follow-up works, including TR Rosetta, which was from the Baker group, which was sort of, added additional ideas like angular information in addition to just distance information. Now, just sort of before this period, around early 2018, myself and another group, actually from W. Marks group at Harvard, developed what I would characterize as sort of the first set of end-to-end differentiable approaches. So the idea here was to there were actually two ideas here. First, to try to set aside coevolutionary data and go to sort of this intermediate state, this idea of using position-specific scoring matrices, where we essentially look at the individual residues one at a time and look at their propensity for mutating to other residues, but don't explicitly model inter-residue interactions in terms of the sequence information that's coming in. That was one idea. And the other idea was to actually try to kind of formulate this whole approach in an end-to-end differentiable manner, so that you are going from sequence to the final predicted structure without any kind of physics-based engine, without any kind of post-processing step. And there are good reasons for why one wants to do this, and I'll come back to it in a moment. But these were sort of the first methods, as I'm kind of depicting here. They didn't necessarily work better than existing approaches. In fact, they were not really competitive with the coevolution-based approaches here, but they were, I think, kind of provocative in terms of what they were able to do. And they did have the virtue of being much, much faster, on the several orders of magnitudes faster than existing approaches. NEMO, actually, this other approach, even tried to go from single sequences, but not surprisingly, sort of suffered a performance penalty due to that. Now, fast forward to 2020, RaptorX sort of kind of continues to build on these basic ideas, and actually, as Jim just described, goes back and tries to do things from a PSSM. So again, kind of trying to set aside all this coevolutionary information and see if we can kind of push into sort of the harder direction. In the opposite direction, now we have AlphaFold 2, which really says that, all right, well, we're going to throw the kitchen sink at it, and in particular, do it in a way where we're going to take these MSA objects, treat them as essentially raw objects, right? Not try to extract any summary statistics from them, with the aim of potentially capturing, one can imagine several things. For example, higher order correlations, right? So triplets or quadruplets of residues, for example, but also maybe more sort of global aspects of sequence evolution. For example, phylogeny, right? These are things that are not easily extractable from sort of pairwise correlations between residues and sequences. AlphaFold 2 is definitely, I think I'm actually underestimating how well it's worked. It's really sort of, I would say a little bit higher up on that y-axis. I think it's nearly solved the problem, basically, at least for the multiple sequence alignment case. And this was also an end-to-end differentiable approach, and I think that's one of the key ingredients that made it work as well as it did. But what I'll tell you about today is what I sort of consider as the holy grail in a way, which is really being able to work as well as AlphaFold 2 does, but do so from individual protein sequences as opposed to any kind of evolutionary information. And the work that I'll describe, that's still unpublished, is in that vein of trying to kind of push in that harder direction. Yeah. So this is sort of a high-level kind of map of where things currently exist. But let me kind of decompose this sort of algorithm space into kind of the various components that seem to kind of essentially make it up. Although one can imagine sort of other architectural motifs as well.
So Mohammed, I'm going to editorialize for a second and basically say that in a way, what's happening inside the cell doesn't have access to any of these three columns on the right. We know that the cell is solving the problem on the left. So basically from first principles, you should say, well, a protein, if it can fold in vitro, it should be able to sort of do this and the information should be there. But my question to you is, can it actually fold in vitro? In other words, how much of the chaperones inside the cell are in fact guiding that, and how much information is actually even there in the primary sequence of these proteins?
Right. So that's a very good question. So I think you're right on both accounts, right? In the sense that, if we could do this from co-evolutionary information, whatever, we should be able to do it from sequences because, in principle, at least information is there. We know the cell does that, right? But on the other hand, we sort of also know that it doesn't because there's also additional information. What I suspect, so there's two views here. One view is to be very purist and say what I really want is something that is like a physics engine that would recover essentially the lowest energy state, given this polypeptide chain in solution. If that were the problem, then yes, I think you're right. To be able to actually fold a protein, you would in fact have to really reflect the cellular environment, including all these other cofactors and post-translational modifications and chaperones and so on and so forth. However, what we do see, and so if we're not to be very purist, and say, all right, I still want to be able to predict from individual sequences because that's a useful thing to do. There are good applications for that. But I want to still get the native state without necessarily kind of recovering or requiring all these physical aspects to be present. I think that may still be possible, and AlphaFold 2 is an indication of that. So there are several cases there, several examples in the AlphaFold 2 kind of predictions, which were interesting because, for example, I actually have some slides at the end, but I'll just kind of tell you. They for example, predict an active site where, with all the side chain atoms in essentially almost perfect position, even though that active site is actually stabilized by, I think it was a zinc ion. But they don't model zinc ions. Yeah, they're just predicting sequences.
Yeah.
But presumably because they've seen enough crystallographic structures, they sort of know that particular kind of orientation, that particular set of residues really like to be oriented in that way. And in a crystal structure, there's going to be a zinc ion, but I'm just going to kind of impute it effectively.
Now, a related question, which is, I think, related to your Holy Grail question, is if I want to predict the impact of a protein-altering mutation in a new individual, or that happens in cancer, or something that no one has ever seen before, will AlphaFold 2, for example, fail? Or will there be enough information still captured in the primary sequence to sort of say, oh, wow, this will dramatically change the structure? Or because it's constrained by so much other information, if it's missing an ion, for example, it still finds it, will it sort of care about a mutation?
So I think we don't know because it's not really been tested in that way with that sensitivity. I think, a year ago, I would have said probably not, because these things are very MSA based and they acquire, hundreds, thousands of sequences. So it's really all about the sort of averaging that's happening across the family. Now I'm not so sure because of what you just said. And particularly because the way they set up their structure module, which is really operating in 3D space, and as far as we know, is just operating on the structure. So it doesn't have any additional information. So it may actually be learning some implicit physics in doing so. So there may be enough information there, enough constraints for it to actually detect, what a single mutation would do. But that certainly has not been shown. It sort of remains a question. And I do think it's going to be an interesting research direction for the next at least couple of years, being able to kind of take these methods and apply them in these sort of various, say, application areas, which require sort of resolution in a fairly directed way, right? In terms of like specific regions or specific type of disordered parts, or or for example, mutations in cancer.
Perfect. I love it. Thank you so much. Carry on.
Cool. Thank you. All right. So coming back to this sort of algorithm space, or rather the decomposition of the algorithm. Like I said, there's sort of kind of three main components. There's the input, this is what goes into it. There's the torso, this is the thing that does kind of the reasoning, the typically neural network. There's the output, the thing that you're generating. That could be the 3D structure, but for the most part, up until AlphaFold 2 and some of the work that we've done, that typically was some sort of proxy object, something that was not the structure, but that is related to the structure. And then that was then fed through a post-processing pipeline to generate the final three-dimensional structure. And I should say, I'm going to sort of highlight here in the top right, as I describe kind of the various sort of variants of the architecture, the different methods that map to it. Now, I mentioned earlier this notion of end-to-end differentiability, and sort of one of the things that I think we sort of innovated in. And the reason why that's important, right, is the following. So the output, the thing that you generate from the torso, is the thing that gets optimized in the machine learning framework, right? This is where your loss function is defined, and this is what you go into essentially do gradient descent on, right? So if you're optimizing on some sort of proxy quantity that's sort of distal from the thing that you want at the end of the day, then you're sort of leaving something on the table. Right, and that's fundamentally kind of the value proposition of end-to-end differentiability. And really, I would say much of what sort of deep learning has achieved in the last decade is sort of taking these kind of disparate components and then stitching them together so that you can optimize the whole thing jointly from beginning to end. Right. So within this context and kind of this idea of end-to-end differentiability is that this kind of the green, yellow, blue part is the part that is end-to-end differentiable, and everything else beyond it is not. So coming now to the method that I'm highlighting in the top right, and just to kind of pin them down in terms of this decomposition, on the input side, like I said, you take a multiple sequence alignment and you're extracting essentially pairwise correlations from those residues. That then sort of gets converted into a contact map, a binary contact map. At least initially, this was the case. So it's basically kind of a filtering, a denoising approach that takes a sort of messy thing and cleans it up to something that resembles the ground truth more closely. With AlphaFold, with RaptorX 2018, this included continuous distances, not just discrete distances, and then TRZ actually introduced the idea of including orientational information as well. Same general paradigm, but additional types of data. And all these approaches basically kind of relied on this idea of post-processing, typically using some sort of Rosetta-based pipeline, actually, because Rosetta tended to be kind of the most, let's say, kind of the most sophisticated or the most mature in terms of doing this. And even AlphaFold 1 actually relied a bit on Rosetta to make his predictions. Now, one thing worth noting here is that this can be quite slow, right? Hundreds to tens of thousands of hours, depending on how extensive this post-processing is. Parenthetically, AlphaFold 2, as far as we can tell, does very little post-processing just using Amber, but actually takes quite a bit of inference time. I can talk about that, enormous amount of inference time, a little bit unclear why, but sort of funny in that they got rid of the post-processing, but the neural networks end up being super slow. So there you have it. But going back to this point, right, the issue of post-processing that prevents optimization of the final structure. So that's a point I made earlier. Another kind of technical point, right, is that these things that are being created here, right, in the way they're currently formulated as far as distances or orientations, they inherently have the potential to be self-inconsistent, right? Because this is really an overdetermined object. You don't actually need all these distances to determine how a set of points is going to be realizable in a Euclidean space. And so because you're sort of overdetermined, you could essentially be making predictions that are self-inconsistent that cannot be embedded in 3D space, right? This is a basic kind of geometric fact. And that's in some ways kind of the task that was left to the post-processing, is to take these potentially self-inconsistent predictions and then make them self-consistent, get a three-dimensional structure out of them. So now I'm going to kind of shift to the RGN and NEMO, so the work that we did and others as well. And here again, the basic idea is say, okay, let's actually predict the final three-dimensional structure, right? Let's not introduce these kind of proxy quantities. And in particular, what this means is that because we're predicting the final 3D structure, then self-consistency is going to act as an additional constraint over the model, right? And that's actually kind of nice, because that means that your neural network is going to basically have to behave in a way that sort of never makes it kind of generate nonsense. And any additional constraint that one can place on the model likely improves its performance, right? So it's always a win when you can place additional constraints. So when we set out to do this, right, the dream was to predict a single sequence, to use a single sequence to make a prediction. That didn't quite work, and I'll mention that in a moment. But the way we kind of formulated this was the kind of the original RGN, the first model, was to basically parameterize the local geometry around any residue using a sort of a discrete alphabet of torsion angles. So the 5 psi omegas here are corresponding to the sort of the Ramachandran angles essentially, and you essentially have a kind of a discrete alphabet at any residue position, and then what you do is you predict a probability distribution over that alphabet. So you're kind of picking from that discrete distribution, but we don't sample from this distribution because we want to maintain end-to-end differentiability. And instead, what we do, and this gets a little bit technical, so I won't delve into it too much, but basically we kind of project those angles into the complex plane, and then sort of do a mathematical trick that basically treats these as if they were in Cartesian space, even though they lie on a circle, extracts sort of a mean coordinate, and then pops that back onto the circle. So essentially, it's a way to extract an angle in sort of almost like a mean field approximation from this distribution of angles. We think now this can be done in a much more mathematically sort of principled way and in a physically better way, but at least this is how we did it the first time around. So having done so, we now have these sort of free torsion angles at every iteration or at every residue, and then we simply construct the structure basically iteratively, so autoregressively, if you may. So at every residue, we predict the torsion angles, we rotate based on that torsion, we predict the bond rotation, we rotate based on that rotation, and then we do bond extension, and then we extend based on that. And then this process just is repeated over and over again, essentially kind of recreating the chain one atom at a time. It's not clear what AlphaFold 2 is doing in terms of the end-to-end differentiable approach, how they're constructing the structure, because they are going from this kind of histogram picture to the 3D coordinates, and they didn't really describe that process. They may be doing something autoregressive like I'm describing here. They may be doing something else. That sort of remains to be seen. But this is at least how we sort of formulated it up.
Basically, you're focusing so much on the protein backbone. But in fact, there's so many interactions between these residues that you seem to not be modeling, in the sense that after a first few atoms come in, or like residues come in, amino acids come in, they start interacting with each other, and then that shapes the original structure. You seem to sort of have been making each decision and be done with it.
So that may partly be true. It's not entirely true. And let me just sort of point out a subtlety here. Right? So the loss function is defined on the full three-dimensional structure, right? The loss function is not defined in terms of how well we're matching these angles. If that were the case, this would never work, precisely because of the point you mentioned, right? Because so local accuracy does not result in global accuracy. But the loss function is defined in terms of the global accuracy. Now, it is true that the prediction, at the end of the day, you're predicting each torsion one at a time. But by the time you've gotten to that stage, there's been multiple layers of, in this case, of a recurrent network that's sort of already seen the internal representation. So what I suspect has been happening is that internally there's sort of implicit homogenization of the structure, almost like an implicit histogram or something like that that's happening, so that by the time it's making its predictions, it's sort of already resolved these issues. But this is a hypothesis. We don't really know how it's working. But the kind of what we do know, or what is the key point, is that while the structure is parameterized in terms of torsion angles, the actual loss is global.
Can you observe the training process to see what is getting optimized, or is that too involved?
Yeah, you could. Yeah, actually, I used to have an animation for that. I don't have it in these slides. It was sort of interesting. I mean, it seems, at least if one sort of stares at it, that initially it sort of learns fairly local geometries, fairly local sequence structure correlations, in the beginning of training. But then as time goes by, it starts learning kind of more global aspects and start putting kind of pieces together. So that does seem to be the case. And actually, AlphaFold 2 seems to be doing something similar. So doing their talk in CAS 14, if you look at the histograms that they form internally, it starts out about sort of local structure, but then over time it starts capturing in the case of a multi-domain protein, sort of inter-domain interactions even, which is quite interesting. So it goes from local to global.
Thank you.
Yeah, you're welcome. Right, so this is how we sort of parameterize output. And then I sort of said earlier, at that point in 2018, we didn't quite get to the point of single sequence. So we essentially went to these PSSMs, which generate or summarize distributions over individual amino acids. So it's sort of a compromise between the single sequence case and the kind of coevolution case. The torso, there's a question of what torso to use. At the time, we use a sort of you can use a convolution network, you can use a recurrent network. I think it's pretty clear now that transformers is the way to go, but at least for this architecture at the time, it was a recurrent architecture. So just to give you a sense, this is one of the predictions we made at the time. It was sort of, I would say, kind of an intermediate prediction, sort of the median of the distribution. So, they see the helical region is reasonably well predicted, but the C-terminus, not so much so. But also the kind of the secondary structure, the alpha helices themselves are not really particularly well predicted. They're sort of overly wound in the predictions we were making here. They're essentially sort of non-physical, and that had in part to do with the way we parameterize torsion angles that kind of led to this suboptimal kind of pathology. But that was at least how things looked at the time. All right, so now come back to this picture. Let me tell you what we've sort of been doing since then. So we've taken this idea of the kind of torsion parameterization and evolved it a little bit using this construction called the Fourier-Fourier construction, a fairly basic idea coming from differential geometry, where I won't go into the detail, but let me just kind of say the high level point, which is that basically now we only focus essentially on C alpha, so we don't actually bother with sort of all the backbone atoms, and we parameterize it using essentially these sort of rotation matrices that actually simplify the math quite a bit and make this much simpler, both just computationally, but also just in terms of how you formulate it. Because it's essentially to extend the chain now, all you need to do is a series of matrix multiplies instead of kind of the complicated procedures that I was describing earlier. And this has the virtue of solving the problem of this pathological secondary structure. This essentially ensures that the secondary structure we generate in terms of alpha helices and so on is physical. It sort of reflects kind of what we know about proteins. So that's one piece. But the key piece, really, maybe the most important piece, is now we've actually gone back to the idea of a single sequence. But the way we do so is by feeding it through a language model. Jimbo mentioned the MSA transformer and ESM1B from Facebook. Those have been really spectacular in a way. And we sort of in parallel have developed a very similar language model. I just show, if you haven't seen GPT-3, these are these methods where you give them a short prompt and it just kind of goes on giving you flowing text for pages and pages. So it was a similar idea here, quite similar to the ESM1B model from Facebook, but some slight differences. So the basic idea is that you take a sequence, you mask certain residues, you feed it through a series of transformers, and then you try to recapitulate the original sequence. Like I said, and then the idea is that now what we're going to do when we do the prediction is that we're going to embed each residue in terms of its latent space in this language model, and then use that as the input to make our predictions. Some differences here from the ESM1B approach, we don't just drop individual residues, we also drop fragments, because we think that sort of challenges the model more, and that maybe represents some of the things that you see in biology. And we also do this trick where we could splice in two different proteins, and then we ask it whether those two proteins, whether the sequence is sort of an unperturbed sequence or a mixed sequence. And that kind of challenge we think also improves training performance.
These are very cool tricks. I love the concept. And we talked a lot about that in our representation learning lecture very early in the term. I'm curious, have people been sort of creating these types of tasks where instead of sort of having to have a bunch of solved, folds, you're forcing the system to learn these representations by having these tasks that allow a lot of, almost self-supervised learning?
Yeah, I think it's actually very, very much under-investigated. There's been a lot of work on the kind of core problem, right? And just basically taking ideas from NLP and applying it here and scaling it. And I think that's what the Facebook models have been really good at. But there's not been that much innovation in constructing new tasks that kind of really leverage the biology of proteins, right? And or even just kind of new tasks, period. So I think this is a fairly promising direction to pursue, and just coming up with better ways to train these models.
Yeah, I love your idea so far. This is great.
Thank you. So all of them describing the architecture. So one kind of thing we did do here, a bit of a compromise, was to come back to do some post processing because it improves performance. Basically, that's the only reason. We can still make predictions, and we still train the model end-to-end differentiably for the initial structure, but we do add additional post processing step. Like AlphaFold 2 seems to do, because that improves performance. And this is a bunch of kind of Rosetta magic to do so. I want to dwell on it just for the sake of time. So let me just show you some results now, and then I'll wrap up. So in terms of RGN1 versus RGN2, this is one target, Cas14. So I'm going to show you results from Cas14 here, which is the one that just happened. So this is sort of interesting. So this is a TROZ ground. This is the ground truth because at the time we didn't have the ground. I don't think we actually had the structure yet for this one. So this is what TROZ predicts. We have the RMSD because CASP tells us what the RMSD is, even if we don't have the structure. So the RMSD is with respect to the ground truth, even though we might not have it. Oh, I'll actually, no, sorry, here we do have it because I see it in white. So here we do have it. Okay. So so this is the original RGN prediction, quite poor. RGN2 prediction, you see, fairly significant difference already, halving the RMSD. And then this is pre-refinement. Then when we do refinement, we bring that down significantly, down to almost within spitting distance of the TROZ prediction. Although the speed up that we have with the end-to-end differentiable approach, we lose a lot of that because of this refinement step. Now, just one point, so I don't hop on this repeatedly. Keep in mind, this is not being made from a multiple sequence alignment. This is based on a single sequence that has gone through that language model. Right? So this is the key thing here, where accuracy is not quite as good as the state of the art, but we're using a lot less information to make that prediction. Let me show you some admittedly cherry-picked examples against AlphaFold 2. I'll show you some of the best examples and some of the more middling examples. So these are all predictions aligned with respect to AlphaFold 2. Here again, we don't have the ground truth, so we don't quite know how well the real thing looks like. But the RMSDs are with respect to the real thing because CASP tells us what these numbers are. So that gives you a sense. So these are, in the best cases at least, we are, within spitting distance of AlphaFold 2, even though we're using individual sequences and they're using full MSAs. A more realistic set of examples like these. Here you see a much larger gap between us and AlphaFold 2. This one is sort of interesting in a way because neither is actually doing particularly well. I mean, in principle, we're doing a little bit better, but I think they're both pretty crappy. What is interesting here, though, this seems to be a monomer that's part of a multimeric beta barrel. And AlphaFold 2 is predicting this sort of very stretched thing. We're predicting something kind of more globular. We don't know the ground truth, so we don't actually know what the thing looks like. But it's sort of an interesting example where, at least in this region, we seem to be in agreement with AlphaFold 2, but here is sort of where we deviate. And if we do have, we do have some cases where we do have experimental structures. Those are some of them I can show you here. And so you see, in some cases we do okay, in some cases we do so-so. On the whole, we're certainly not anywhere near the kind of the coevolution-based approaches in a systematic sense. But we have wins in some cases. However, let me skip this for the sake of time. The one area we do actually do well systematically is in two various regions. One is singleton proteins, and one are de novo proteins. So what I mean by singleton proteins are proteins which are essentially in kind of the twilight zone of sequence space, which have no sequence homologs at all. So what we basically did, we took the unique class 30 data set, and we identified sequences which seem to form their own clusters, meaning they have nothing else in that cluster, at least within a 30% sequence identity ball. And then we assess the predictions on this on our method versus TROZ in this case. AlphaFold 2 is not yet released, so we can't assess against it. So in this quadrant is where we're looking at two metrics, RMSD and GTTS. In this quadrant is where we do best on where we do best on both metrics. In this quadrant is where we do worse on both metrics, and here's sort of a split. And for the singleton proteins, I think on 83% of cases, it's a little bit hard to see with this graph, we actually do better than TROZ. So this is really exciting, right? Because this is now single sequence prediction in a regime where you sort of can't use coevolution, and this approach really seems to be making a difference against not AlphaFold 2, but at least a state-of-the-art publicly available system. So that's cool.
This is absolutely wonderful. And my question is, what are these proteins? I mean, are these like some weird exotic bacteria that no one has ever seen before? Do you have any insights for the class as to what these proteins are?
Yeah, I think they're primarily what you said. There are some de novo proteins, even like in mammals or insects or something, because some of these are fairly young, and like if you go off of a tree, there may just not be enough genome sequenced around it, and so you can actually get like a de novo protein, essentially natural de novo that emerged, and that you just sort of caught it without any sequence homology.
There's a related question to that, which is, it's possible that evolution hasn't yet quite optimized the structure of those proteins anyway.
Right, absolutely. Yeah, that's quite right.
A lot of the insights that we have from other proteins might simply not apply here because they're not quite at their stable form yet.
No, that's very interesting. That's very, very interesting. I hadn't thought of that, but yes, that's a great idea. That's a great point, actually. So they may, in some ways, kind of present a distributional shift into the structure from what you would otherwise expect. Yeah, absolutely. So that's one set that we do well on systematically. This is not cherry picked. And the other set are de novo designed proteins. Unfortunately, color scheme is not ideal here. We're working on this, but again, these are the ones we do best on. I think this is about maybe 50% or so. So it's 50% we do best on, maybe 10% we do worse on, and the remaining 40% is sort of a split. What is interesting about these proteins is that they were actually designed using Rosetta. So it's sort of nice that we actually can sort of do better than Rosetta, even though they were designed by the Rosetta energy function to, not TR Rosetta, but the Rosetta energy function to come up with these de novo proteins. And so again, this is another area where it sort of makes sense that these types of sequence-based approaches would make a difference. And of course, I mean, this is probably one of the areas beyond kind of like predicting mutations and so on, but protein design obviously is sort of the natural directions where these methods would be useful. So let me end on this point. So why is this useful? Right? So again, if you don't do refinement, at least there's a rather significant speed up, on several orders of magnitude, which might be useful for things like, say, docking, right? Especially if you want to be conformationally flexible, where you predict different protein conformations based on, say, where a small molecule lives. Things like Manol's mentioned, like genetic variation, right? If you want to be able to predict how mutations change things, maybe in principle having single sequence-based methods would work better, but we don't know yet. And that's the proof will be when AlphaFold 2 or some version of it is released and we can actually compare head to head.
But again, it might not be necessarily can I predict it from scratch, but knowing that initially it was this, can I sort of see what the impact on the structure? And that might be a whole other problem altogether.
Right, exactly. It's almost like a refinement problem. You predict the general fold using an MSA, and then you kind of refine it into the actual structure using another stage. Right. Rapidly evolving viruses is another example where you have things that are just changing very quickly, and it's really hard to sort of keep track of them. And obviously, protein design, like I just said, those are all areas that I think would be quite exciting.
Suppose hypothetically that there was this crazy new virus that has 3 parts of a spike protein from 3 different relatives. I mean, that'd be crazy, right?
No, but absolutely, that's exactly the kind of thing which this would be hopefully very useful for, right?
Completely hypothetically.
Yeah, completely hypothetically. Yeah, so that's it. I just, I actually mentioned that the people who did all this work, Ratul and Nazim and Serge. Ratul and Nazim are currently still at Harvard because I was there and I moved to Columbia. And then Serge is doing his own startup right now, where he was a student with in the George Church Lab.
So I want to ask all of the students to join me in really thanking Mohammed for an amazing lecture. And thank you, Bruno. Thank you, Jinbo. I mean, we are so privileged. And again, speaking of that crazy rapidly evolving virus, there's horrible downsides to the lives lost and the livelihoods lost and the space between the people who should normally be close. But then we're taking advantage of the fact that people who should normally be far can be coming closer together. So thank you for coming from 3 parts of the world and joining us. This was super illuminating. Thank you, all 3 of you. And I look forward to many more interactions. So for Jinbo and Mohammed, sometimes we ask if the students are interested in final projects in your space. Would you be interested in potentially co-supervising them?
Yeah, of course.
Awesome. Beautiful. Awesome. Thank you all 3. And then can I ask the TAs to please stick around? Thank you, everyone. And thanks to the class for staying 27 minutes over time. So that shows how interesting all of these topics are and how interested the
Take care. Bye.
Okay. Bye.
Bye.